We'll continue our discussion on cancer to talk about the way cancer develops. Cancer development is called carcinogenesis or oncogenesis. Malignant transformation is the changing of a normal cell into a cancer cell. And like I explained in the last lecture, this usually occurs through the loss of some aspect of cellular regulation. During malignant transformation, initiation is when oncogenes turn on excessively. They just keep telling the cell to keep dividing. In this stage, it could either be those oncogenes or the suppressor genes. So either way, the result is the same. Either the oncogenes just keep going and they keep turning on, or the suppressor genes are damaged and those oncogenes just kind of keep going with no control. This can cause a change in expression, and it can be caused by anything that can invade a cell's nucleus or mutate the cell's DNA. So this initiation could be due to just about anything that can get into a cell's DNA. This is irreversible and can lead to cancer development if the event or protein or virus or whatever event that caused cellular regulation to be depressed or changed, whatever that event is, if that continues, it's going to just keep dividing and keep getting bigger. And so from this stage, you can see how cancer can develop from just a single cell. We've talked about carcinogens a couple times, and this can be where carcinogens can come in. A carcinogen is a substance that changes the activity of a cell's genes, causing that cell to become a cancer cell. This can be chemicals, a physical agent, a virus. So these carcinogens damage DNA, causing that suppressor gene essentially to fail. So that suppressor gene fails to control the oncogenes, causing the excessive expression of the oncogenes and continued cell division. Promotion is the next step of malignant transformation, and promotion is the enhanced growth of a dividing cell. Promoters are the substances that do this. So once a normal cell has been initiated by a carcinogen, it's considered a cancer cell. At this point, it can become a tumor if it's enhanced. So enhancers can be things that we normally see in our body, like certain hormones like insulin and estrogen. So if a cell has been initiated, so essentially turned into a bad guy by a carcinogen, and then it gets enhanced by something totally benign and something totally normal like insulin or estrogen, it can become a tumor. At this point, we talk about latency period, which is the time between when a cell has been initiated, so when it's come in contact with that carcinogen and the DNA has been changed, and when it becomes a tumor. That is the latency period. So latency period is the time between when a cell has been initiated and when it becomes a tumor. Progression is the next stage of malignant transformation, and progression is exactly what it sounds like. It's just the continued change of the cancer. It usually makes it more malignant. At this point, we're seeing angiogenesis, or the tumor actually creating its own blood supply. And at this point, we're seeing that anaplasia where the new cells are forming different than the original cells, and they don't look the same. And that anaplasia also causes them to have advantages that those original cells wouldn't have had. These advantages can allow them to do things like live outside of their normal area, and then continue to divide no matter what kind of conditions they're in. Metastasis is the next aspect of malignant transformation, and it occurs when the cancer cells start moving. So they move from that primary tumor by breaking off and forming remote colonies. So when they do that, when they break off in metastasis, that's when we see these secondary tumors. So primary tumors are identified by the tissue from where it arose, so from the parent tissue. Depending on where that primary tumor is, it could have a huge effect or it could have not much of an effect. For example, breast tumors, a primary breast tumor doesn't really have a huge effect on the function of the body because the breasts don't form any life-sustaining functions. So it's just not vital. So if a tumor grows in the breast, it's not going to cause death. It's the metastasis of that tumor that is deadly. So if we have a tumor, for example, in the heart, which is unlikely, but a heart or brain or kidney or one of the vital organs, that is when we start seeing more problems with a primary tumor because it's going to affect the tissue or the organ's vital function. Secondary tumors are, like I said, when a tumor metastasizes and the cancer cells move from the primary location to a different location. This is a kind of a difficult aspect of secondary tumors because a secondary tumor is still considered a tumor from the original tissue. So for example, if you have breast cancer, it is your breast cancer that metastasizes to your lungs. You don't suddenly have lung cancer even though a secondary tumor is found in the lungs. So we always name it from the primary location. Here are some steps of that metastasis. Tumors secrete enzymes and that opens up that surrounding tissue and it helps get those cancer cells moving around. Bloodborne metastasis is going to be when the tumor cells release into the blood. This is the most common cause of metastasis. So a tumor is going to secrete enzymes into the vessels, and the tumor is going to be able to, because of its loose adherence, unlike a regular human body cell, its loose adherence to one another is going to allow those cells to break off and go through the blood supply. At this point, tumor cells, metastatic tumor cells, can continue to spread. They'll go through the blood supply and lodge in capillaries where they can move into tissues or damage vessels. And this is usually where we'll see secondary tumors arise, kind of where the capillary stops the cancer cell and it kind of sticks. Table 21-2 in IGGI 8th edition on page 363 is called Common Sites of Metastasis for Different Types of Cancers. That's an important one. So not only do we have bloodborne metastasis, we also have lymphatic spread. And this is going to be a similar process, but this is in sites that are more vulnerable to lymphatic spread. Another figure I'd like you to take a look at is figure 21-5, the steps of metastasis in IGGI 8th edition, page 363. And that's this figure that we have here.